Today's talk really reports on our current efforts to image non-invasively cells of the immune system and the tumors they attack. This work is done primarily by three postdocs, Mike Dugan, Jess Ingram, and Mohammed Rashidian. And we collaborate with Stephanie Dugan at the Dana-Farber Cancer Center. The fact that there are two Dugans on this slide is no coincidence. We collaborate with Ned Keller and Ralph Weislater for some of the imaging stuff. And many of the reagents that we create would not have been here had it not been for the involvement of Steve Almo, a crystallographer at Albert Einstein, who's provided us with purified proteins against which we can raise antibodies. And then last but not least, we're grateful to the Koch Institute. Through a philanthropic gift, we've been able to acquire a PET-CT instrument, which is run by the small animal imaging facility headed by Scott Malmstrom. And some of the images that we have been producing of late are the product of those efforts. So I think this is a wonderful opportunity to advance non-invasive imaging of immune cells at MIT. These are other collaborators, two juvenile alpacas. We immunize these animals to generate antibodies that have unusual characteristics. And one of the reasons why we're interested in these camelids is the fact that in addition to the conventional two-chain antibodies, these animals produce heavy-chain only antibodies. And whereas the conventional antibodies can be shrunk proteolytically into FAB fragments, and with some diligence turned into single-chain FV fragments, the heavy-chain only antibodies can be readily shrunk into the variable regions of the heavy-chain only, and these are therefore the smallest antigen-specific recognition modules to which we have easy access. As some of you will know, the single-chain FVs have been used to construct chimeric antigen receptors on T cells for adoptive T-cell therapy. One issue that confronts the folks who work on these CARs is the necessity of carefully designing the linkers with which the variable regions are connected to ensure stable expression. So why do we use single-domain antibody fragments? Well, their small size, as indicated here, is a key advantage. And their small size leads to improved clearance from the circulation. As you will see, this is important to achieve proper signal-to-noise ratios in imaging experiments. Their small size likewise affords superior tissue penetration. They're a tenth the size of a full-sized antibody molecule. And this is one of the reasons why for imaging applications, full-sized antibodies have not met with great acceptance. They're easy to manufacture. Many of the single-domain fragments that we work with require neither glycosylation nor disulfide bonds. And this means that they can be readily expressed inside cells with retention of antigen binding capabilities. And in E. coli, yields of up to 50 milligrams per liter may be obtained. Equally importantly, and in comparison with some of the alternative scaffolds that have been randomized to create high-affinity reagents, we deliberately and repeatedly immunize alpacas to take advantage of the fact that their B cells undergo affinity maturation just like normal antibody-producing cells. And this yields antibodies of excellent specificity and in many cases of high affinity as well. The best binders that we've obtained have binding constants in the 10 picomolar range. Then, because of their small size and ease of expression in prokaryotic and eukaryotic systems, they're easily genetically modified to create fusion proteins that might be of interest. And as I'll show later on, that same trait is useful to enable chemical and enzymatic modification. And we make heavy use of these enzymatic modification methods for the purpose of creating imaging agents. The goals of this applied branch of work in my lab include attempts to non-invasively image immune checkpoints. We've heard a lot about PD-1, CTLA-4, and so forth. And as has been repeatedly pointed out, one of the things that we must address and that we still do not fully understand is why some patients respond to checkpoint blockade and others do not. If it were possible to predict responses or stratify patients into responders and non-responders, much would be gained. So we would like to be able to predict the responses to these checkpoint blockades as one of the goals of our work. And then finally, having established that some of these single domains can successfully target cell types of interest, can we use this targeting information to deliver useful payloads to the very sites to which these single domain antibody fragments are targeted? That might include radioisotopes for radiotherapy, small molecule drugs as cytostatics, chemokines or cytokines to activate lymphocytes on site. And so one of our goals is therefore to engineer the tumor microenvironment as a means of enhancing favorable immune responses, not only against tumors, but also perhaps infectious agents. The modular possibilities afforded by these single domains are, in essence, limitless. The heavy chain only antibodies, as I've pointed out, can be shrunk to their variable regions of the heavy chain only. And then using either genetic or chemical methods, we can create bispecifics, either with two different antigen binding modules or homodimeric binders that contain the exact same binding units. We can attach FC portions at will and thus create chimeras that have an antigen recognition module hooked up to an FC portion that might enable effector functions. And then equally attractive to us is the possibility of creating fusions of single domains with cytokines for targeted delivery of cytokines that would activate T lymphocytes on site in the tumor microenvironment. We have a pretty expansive collection of single domains in hand. The ones that I'll mention today include an antibody called B3. This is a reagent that recognizes mouse PD-L1. We have a reagent called A12, which recognizes both human and mouse PD-L1. H11, an anti-CTLA4 reagent, and which I will not have the time to describe, are experiments that deal with a blocking antibody directed against the don't eat me signal CD47. It's perhaps worth pointing out that the affinities with which these single domains bind to their murine targets exceeds that of the widely used commercially available antibodies, and that further enhances, we think, the value of these types of tools. Now, one of the attractive features of single domains is not just the list that I just gave you, but also the fact that they often serve as crystallization chaperones. So for proteins that are difficult to crystallize, inclusion of a single domain in the mix often leads to successful crystallization and improved resolution of the crystal structures obtained. So in this particular example, this is done in the lab of Steve Almo at Albert Einstein. We have here the CTLA4 ectodomain in complex with the anti-CTLA4 antibody. And based on the exact site to which the antibody fragment binds, we could already predict that this would be a blocking antibody. And in fact, for the commercially available humanized antibodies, I'm not aware of the fact that the actual structure to which they bind has been published. Now, the modification strategy that we use to equip single domains with various payloads is not just genetic fusions, which would be the most straightforward way of achieving them, but we also use enzymatic modifications, because each of the single domains that we clone and express comes equipped with a recognition motif for a bacterial enzyme called sortase. So if this were our single domain protein of interest, the target, there's a 5 residue sequence appended at the C terminus, LPXTG. And this sequence is recognized by a bacterial transacylase called sortase A. This enzyme is usually involved in the attachment of proteins to the bacterial cell wall. An example that you might be familiar with is protein A, the immunoglobulin binding protein produced by Staphylococcus aureus. It is attached to the cell wall in exactly this sequence of events. So the sortase enzyme cleaves between the threonine and glycine residues with concomitant formation of this covalent acyl enzyme intermediate. And if one now replaces the natural substrate of sortase with a short synthetic peptide minimally composed of a single glycine to which one may append any payload of interest, the transacylation reaction results in the formation of a perfectly good peptide bond via which your payload of interest is now attached to the target protein. It's important to point out that these reactions are quantitative and absolutely site-specific. So with our input single domains, we can convert 100% of the input material to the desired final product. And we shall use this particular strategy to modify our single domains for the purpose of imaging. I've mentioned the fact that we would like to create fusions of single domains with cytokines. Equally importantly, in my view, is the ability to attach chemokines to single domains. And here we face a slight complication, namely the fact that most chemokines require a native N-terminus for them to bind to their G-protein coupled receptor. And we were fortunate enough to collaborate with the lab of Chris Garcia, who solved the crystal structure of one such chemokine receptor in complex with a chemokine and a single domain made by Jess Engram. This structure reveals that the N-terminus of the chemokine protrudes deeply into the bundle of these 7 helices, explaining the need for this native N-terminus. So this is important because it means that one cannot fuse a chemokine to the C-terminus of a single domain, which would be the most attractive position, but rather one would have to attach it to the N-terminus or create a different method of making the fusion. And using chemistry that I'll describe in a little bit, we can in fact modify the C-terminus of a chemokine with a click handle. We can modify the C-terminus of a single domain with a complementary click handle, and then execute a simple chemical attachment that leads to a C-to-C fusion, which would be a genetic impossibility. So this is the chemistry that we use to create our imaging agents. In this line A, we have a single domain antibody fragment equipped with this sortase motif. In a sortase catalyzed reaction, a tetrazine is installed. This product is perfectly stable. It is not radioactive. The reaction itself does not inflict collateral damage on the antigen binding site. And now we use this as a precursor to create, in our case, an F18 labeled derivative. This is done by starting with this tosylated triencyclooctene, which is readily converted into its F18 labeled counterpart. And upon mixing of these two at neutral pH, within 20 minutes, one observes near quantitative conversion into the triazole, which now installs this F18 site-specifically at the very C-terminus of our single domain. It's important to appreciate the need for this rapid process because the half-life of fluorine 18 is about 110 minutes. So as one executes these reactions, the isotope decays under one's very eyes. If you don't want to use F18, but instead rely on a slightly longer-lived isotope, such as copper-64, half-life 12 hours, or zirconium-89, half-life 3.3 days, one can install metal chelators and achieve pretty much the same outcome. Using a sortase catalyzed reaction, the chelator is installed, and then simple mixing with the radioactive metal allows the production of the PET agent. Now, in results that we've published, we have shown that making antibodies against class 2 MHC products allows us to reveal lymph nodes in living mice with acceptable precision. We like to use genetic controls to validate the specificity of each of the single domain antibodies that we produce. So on the left, you see a PET image collected from a mouse that is deficient in expression of class 2 MHC products. And on the right of it, you see the counterpart, a wild-type C57 black 6 mouse. These bilaterally symmetrically distributed structures turn out to be the lymph nodes, altogether absent in terms of labeling from the class 2 knockout. The panels on the right show combined PET-CT images, H and I, through 2 virtual slices at different levels through the animal. Cervical lymph nodes, axillary lymph nodes are visible, and in J, a projection image which shows all of these lymph nodes, including the thymus. If one creates similar slices through the class 2 knockout mouse, in essence, the only thing we observe is background labeling. So the concept we pursue, and it's not the only angle of attack, is that myeloid cells, antigen-presenting cells, neutrophils, and so forth, are often found in and surrounding solid tumors. So rather than relying on tumor-specific single-domain antibodies, we would like to look for the canary in the coal mine. We track the behavior of the class 2 positive cells as an indication of where a tumor might reside. So the first experiment that was done is to take a non-skid mouse and xenograft it with a human melanoma, negative for murine class 2 MHC products. And if we now inject our anti-mouse class 2 MHC single-domain antibody fragment, we can see not only the lymph nodes, but also on the left shoulder, this cloud of label, which represents the influx of murine class 2 MHC positive cells in the tumor. So this is a means of non-invasively detecting the behavior of these myeloid-derived cells. The sensitivity of the method, we believe, is acceptable. In this particular case, it's a heterotopic pancreatic graft, a mouse pancreatic cell line. Upon excision of this tumor, it turned out to be about 1 millimeter in diameter. And in this case, also, we can readily detect the presence of the tumor by virtue of the fact that class 2 MHC positive cells move into the tumor. You may also appreciate that the draining lymph node is hotter, quote unquote, than the contralateral lymph node, consistent with trafficking of immune cells between the tumor microenvironment and the draining lymph node. The competing technology involves labeling of metabolically active tissues with F18 fluorodeoxyglucose. Metabolically active cells more rapidly acquire glucose, and this is a commonly used diagnostic method in the clinic. If we image a mouse injected with F18 labeled fluorodeoxyglucose, the only tissues that light up are the metabolically active normal tissues, and this includes cardiac tissue and the interscapular brown fat. The surprise came when we developed an anti-PD-L1 single-domain antibody and attempted to image the B16 tumor, which in vivo also expresses PD-L1. And this is the result we obtained. The tumor on the left shoulder lights up like a Christmas tree with this anti-PD-L1 antibody, but the other tissue that is positive happens to be the interscapular brown fat. And we would not have known that brown fat is in fact the only normal tissue in the mouse that lights up in this particular setting had we not had access to these imaging agents. You might ask what the relevance might be of having PD-L1 on brown fat. First, let me verify the specificity of this antibody also. On the left, you see a wild-type mouse imaged with the anti-PD-L1 agent, and I call your attention to the interscapular brown fat located there. The PD-L1 knockout mouse, imaged under similar conditions, shows no signal at that location at all, confirming specificity of the agent that we use. Now, brown fat, especially in neonatal mice, is important for thermogenesis. It relies on the expression of a protein called UCP1, uncoupling protein 1. And instead of using electron transport to drive ATP synthesis, electron transport is used to generate heat. And Jess and Mike, much to their amazement and certainly to mine, discovered that juvenile mice of the PD-L1 knockout phenotype have a consistently elevated core body temperature, indicating an important metabolic role for the PD-1-PD-L1 axis in thermogenesis. And we're currently trying to find out exactly what the underlying mechanism is for this metabolic role. I do wish to emphasize the fact that it's unlikely that this PD-1-PD-L1 effect seen in brown fat is unique to brown fat, and I think it's reasonable to assume that the PD-1-PD-L1 axis plays an equally important role in controlling metabolism in immune cells, and this will be another area of pursuit. I pointed out that we can easily make genetic fusions of single domains, and I'll show you just one example of this where we created a bispecific agent composed of our anti-PD-L1 antibody fragment, about which I spoke, in conjunction with anti-CTLA4. The idea here is a very simple one. If we could block CTLA4 on the T lymphocyte and thus relieve inhibition, and at the same time hit PD-L1 on the tumor and block access to PD-1 on the T cell, we should achieve a therapeutic benefit. And in this B16 model, we see tumor growth when animals are given an irrelevant antibody. The single agents do slow down tumor growth, but the bispecific single domain, which hits both CTLA4 and PD-L1, is more efficacious than either of these single agents alone. And I should emphasize here that we have done fairly little to optimize dosing, and so we don't know whether this is in fact the optimum result achievable. Cytokine fusions have been important in demonstrating that having seen the targeting of our anti-PD-L1 antibody to the tumor, we should now be able to deliver activating cytokines to those very same sites. And without going through all the symbols here, I just want to point out that if we administer an anti-PD-L1 IL-2 fusion, we see a therapeutic benefit compared to all the relevant controls, not just in terms of the size of the tumor, but also in terms of overall survival in this very active and aggressive tumor. We've taken a different anti-PD-L1 antibody fragment, the A12 molecule, which cross-reacts with human PD-L1. And in this case, again, when hooked up to interferon gamma, we see a very significant therapeutic benefit in a model that has already been described to you. The combination of TA99, an anti-melanoma efficacious antibody, in conjunction with these single-domain cytokine fusions. And I should thank Monique Kauke from Daryl's lab for helping us with the antibody fragment. The fusion of our anti-PD-L1 with IL-2 also reduces the size of tumor grafts. This is the Panko2 cell line. If one excises the tumor from animals that have received the B3 IL-2 fusion, the anti-PD-L1 IL-2 fusion, there's a highly significant reduction in the tumor weight, and that's also obvious from the micrograph shown below. The number of infiltrating CD8 T cells is significantly elevated when we administer anti-PD-L1 IL-2 fusions. We have yet to determine whether this is expanded proliferation, retention of cells at the site of the tumor, or active recruitment. But again, the result shows a highly significant difference between the IL-2 fusion and the irrelevant controls. And then most recently, and here in the interest of full disclosure, I have to point out that I'm a co-founder of a small biotech company called 121 Bio. They have provided us with an antibody fragment that recognizes murine CD8, and we've treated this in exactly the same fashion as the other single domains that I've just shown you. So this allows us to visualize the distribution of CD8 T cells non-invasively, and once again, all the lymph nodes show up beautifully, and on an appropriately exposed PET-CT scan spleen as well. You may ask what these other strongly staining organs represent. These include the bladder and the kidneys as the main organs via which these single domains are cleared from the circulation. This is a problem that we have yet to address, but in the upper thoracic region, the resolution is really quite good. Can we use this particular method to detect the influx of CD8 T cells in a tumor? These are very preliminary experiments. We have yet to optimize the conditions, but here's an example of a mouse that bears a B16 melanoma on the left flank, imaged with the anti-CD8 agent. Once again, one can see all of the lymph nodes in the upper thoracic region, but now we can also detect the presence of CD8 T cells that move into the tumor. So we think this bodes well for tracking in vivo tumor responses non-invasively, and we are very eager to explore collaborations with our colleagues at the Koch, who have fantastic models for tumor immunology. And then I'll close with two examples from experiments done in collaboration with Vlad Vrbanac at the Reagan Institute. They're very much interested in these humanized mice. The model that Vlad uses are the NOD-SCID mice engrafted with human fetal thymus, liver, and bone marrow. And in these mice, there is a pretty decent reconstitution of both CD4 and CD8 T cells, as well as B lymphocytes. The question is, can one now use human-specific single domains to image the distribution of human immune cells in these humanized mice? So this is an experiment in which the mouse was imaged with an anti-HLA-DR single domain. As I'm sure many of you know, activated human T cells, the CD4 at least, they express class 2 MHC products. And in this BLT mouse, we can readily detect the spleen as a source of these class 2 MHC positive cells, as well as the bone marrow. I think it looks a lot better on my screen, actually. And then what was obvious when Vlad examined these mice is that they suffer from a condition that resembles graft versus host disease. The question we had was, can we detect non-invasively any pathological signs of T-cell infiltration in organs affected in this particular model? And in the BLT mice that Vlad provided to us, there is liver involvement as well as skin involvement. And at least as far as the liver is concerned, we can readily detect the influx of massive numbers of class 2 MHC positive cells. And upon dispersal of the liver, the presence of these cells could be confirmed by cytofluorimetry. So what I've told you is that these single domain antibody fragments are, to us, incredibly useful for a number of different applications. We can use them to deliver important biologically active payloads to the tumor microenvironment. And the goal going forward clearly is to develop these into agents that we can use to re-engineer the site at which these lymphocytes reside. The imaging part to us is important as well, because for the first time we might be in a position to track immune responses against tumors in relationship to checkpoint blockade. Even in the mouse models responsive to CTLA-4 blockade or to PD-1 blockade, not all mice respond equally well. And experiments that we would very much like to do include those that might allow us to stratify mice and predict which ones will in fact respond and show tumor regression versus those that will progress. And then finally, There are two interesting things that are pretty much on the drawing board, but that we will pursue vigorously. One is, can we make chimeric antigen receptors based on some of these single domains? I pointed out at the outset that the single-chain FVs frequently used for the construction of cars suffer from assembly issues. The single-chain FVs are not always of desirable stability because they have never been through the secretory pathway of a eukaryotic cell. The single domains that we work with were, of course, selected in evolution to work in the complete absence of a light chain, and therefore we would predict that these are interesting modules to consider for the construction of these chimeric antigen receptors, and that's work in progress. And secondly, one of the reasons why tumors might not respond to checkpoint blockade is the simple inability of T cells to reach that site. If the tumor fails to emit the proper chemotactic cues to attract T lymphocytes, then no amount of checkpoint blocking will be helpful. In other words, if we were able to first attract lymphocytes to the site of the tumor using a single domain fused to a chemokine, and then hit those T cells on site with a similar fusion that now delivers an activating cytokine, we might be able to improve therapeutic efficacy. So those are experiments that are in the works. And I hope that if this symposium repeats itself, we'll be able to report on those results at a future occasion. Thank you very much.